Hello, students. Welcome back to April Class 1. First of all, I think you are still in your holiday, okay? I think next week you are starting back your school, isn't it? Okay. So today we are going to finish up chapter 5 and then we are going to enter the new chapter of chapter 6. So we have some new students joining us today. And also, I think there are some trial students who join us today as well. Okay, so welcome to the lesson. I think I will leave the discussion of the exercises at the end so that you all can get the knowledge first. Maybe we will discuss about the objective questions, later, and the structured questions and the essay questions later. We start with the discussion of the notes first. Okay, I think that will be more beneficial for the newcomers. So let me just go on to the whiteboard. All right. So for students who didn't attend the lesson last week, I'm talking about the new students especially. Okay, we are currently at Form 5, Chapter 5. All right. So in my course structure for Chapter 5, I will divide it into three parts. So if you attended my lesson, you will see there are three sets of notes in the Google Classroom. One is a note one, okay, two is note two, and three is note three. So in note one, we mainly focus on what is tropism reaction, okay, tropism response versus nastic response. That's note one. In note two, we focus on uh three things, okay. Of course, the first thing we talk about the roles of the phytohormones. So we learn about the different function of the phytohormones. Then from there are two important tropism response that you need to know in your syllabus. One is phototropism, and two is geotropism. Okay, so this will be the basic structure of note 2, in which this was what we focused on last week, mostly on these three things. Note 3 is on application of the phytohormones. So of this, there are two important hormones that we are going to learn. One is auxin, two is ethylene. So last week, we have already talked about ethylene. So today, we are going to talk about auxin first. All right, ethylene is something that we have done already. So we have to talk about auxin today. Okay, and of course, some other applications of other hormones. But of course, the most important ones will still be auxin as well as ethylene. So without further ado, let us continue note 3, focus on the application of auxin in the industry. So let's talk about it. All right. So when we talk about application of auxin, okay, this is a very important exam question. I can be very, very sure some of your school, I can almost use the word guarantee. Some of your schools will definitely come out this question. You will definitely see this question at some point of your life in your Form 4, Form 5. Okay, definitely. So we talk about auxin application. You need to know a few things. Number one, okay, the important one, the most important one is a process called parthenocopy. This is the most important thing that you have to remember. I repeat, parthenocopy. Okay, so in Bahasa Melayu also it's called parthenocopy. Just the spelling is different. There's no R, there's no H, and then it becomes a K, and this is I. That's all. Okay, so parthenocopy is what? Parthenocopy is a technology in which we are producing seedless fruits. I repeat, okay, it's a technique for us to produce seedless fruits. All of us like to eat fruits, for example, like grapes, but we don't like the seeds inside the grapes because sometimes it's It's going to be quite troublesome when you want to get rid of the seeds of the grapes. So a lot of industries, they start to produce seedless grapes. Okay, nowadays you can get it very cheap in the market. You can find those green grapes, big, juicy, and they are seedless as well. Okay, so how they produce those seedless fruits actually is a paradox. That means it shouldn't happen in reality. Why? Because the main function of the fruit is to disperse the seed. If you look at the function of the fruit itself, am I right? So it's for the animals, including humans, to eat the fruits, and because we don't eat the seeds or we don't digest the seeds, we hope to disperse the seed elsewhere so that they can grow into a new plant. That's the purpose of the fruit, actually. So seedless fruits, in nature point of view, is actually useless because they can't grow into a new plant. You should know, basically, the young plants, they are basically in the seed. The zygotes, they are in the seed. Of course, we'll study about that in chapter 6 later. But seedless fruits in nature makes no sense. But in commercial point of view, it is going to sell off well because people likes to eat it. Okay, it has a better commercial value. People prefer seedless fruits rather than fruits that have seeds. So we have seedless grapes, we have seedless watermelon, and so on and so forth. Okay, so the technology to produce seedless fruits is called parthenocopy. And how do they do that? Usually, they will show you a picture. May I know, fruits were coming from where? Do you know? This is just a basic concept. Do you know fruits came from where? Before they become fruit, which part are they actually? Do you know? Anyone knows? Fruits are which part of the plant? Flower, okay, you're right, it's part of the flower. Specifically, is the ovary. Fruit is the ovary of the flower. So what happens is that usually they will draw a flower. For example, I simply draw lah, okay, a flower. So they spray auxin to the flower. So whenever you see this thing, they are trying to perform parthenocopy. So they don't spray on the fruit, they spray on the flower, because the flower, usually they need to be pollinated, then they need to be fertilized, then only they can become fruit. So if there's no fertilization, usually there is no fruit. Just like if the male doesn't fertilize the female, will the female get pregnant? No, right? So the process of producing fruit is like the female get pregnant like that. Okay, so in natural, it will not happen by itself. But if you spray auxin to the flower, what happens is that the ovary will become fruit without being fertilized. So the key word here is that there is no fertilization in this process. Dia tak ada proses persenyawaan. So parthenocopy adalah teknik untuk menghasilkan buah tanpa biji benih. Okay, so proses ini tanpa melibatkan proses persenyawaan. So this is something that is very important. These 3 keywords you must remember in your exam. That's all. I want to keep things as simple as possible. These are the things you need to know. So the next, number 2, another important and famous application of auxin is in the process called stem cutting. In Bahasa Melayu, it's called keratan patang. Keratan patang. Stem cutting. Okay? Keratan patang. All right. So what is the meaning of stem cutting? For example, today I feel like the durian tree, okay, produces very tasty durian. I want to get a breed of this durian tree. So usually what they can do is a process called stem cutting, meaning I get a stem of the tree. So let's say this is the tree. All right. Let's say this is the tree lah. This is the stem. All right. So this is the tree, let's say. So what I do is I will actually take part of the stem. So this is the stem, right? Okay. And then I will poke it into the soil, and then it can actually grow into a real plant. So if you know Cantonese, we call this as bok xu. Usually we just need a stem, okay, and it can grow into a new plant. Okay. Have you heard of this technique before? Stem cutting. Last time I think in KH, Kemahiran Hidup, I think you pernah study this thing also in one of the pertanian things lah. Okay, so if you want to get a new plant, there's a technique called stem cutting, in which you get a stem and you grow the stem into a plant later. Okay, so the problem of this stem is this stem has got no roots one, you know. So how can they actually grow faster, or how can they absorb water and mineral salts without roots? It's going to be very difficult, isn't it? So that's why at this point we can spray auxin to this stem, all right, and they can start to grow small roots around the stem. So you can see they start to grow, okay, small roots around the stem that you cut it out. So these roots, they are not the real roots of the plant, not like the tap root and things like that. No, these are the adventitious roots. I repeat, we call this as adventitious roots. In Bahasa Melayu, we also call this as akar adventitious. Akar adventitious. Later I will show you the spelling, okay? Akar adventitious. So these roots, the function is just like normal roots. They are used to absorb mineral salts and water. So why do we use this in stem cutting? Is because we want the stem to absorb water faster so that they can grow into the original plant better. Okay, so this is very important also. It's an application of the auxin, which is in the process of stem cutting, growing of adventitious roots. Okay, this is point number 2. Point number 3, this is sometimes asked in the exam also. Auxin can be used as weed killer. Weed killer. Weed killer. What's the meaning of weed killer? So sometimes you, like for example, you are staying in a house with yard. Okay, you're staying in a house with yard. So if you don't care about the yard, what happens is that the weed will start to grow. There'll be a lot of the rumput, you know, they grow until very tall, and it'll be difficult for you to do the process of gardening. Okay, so what we can do to kill the weed, in Chinese, these are 杂草, all the rumput that you don't want. Okay, so how we kill the weed is we can also use auxin. This one depends on your knowledge of last week, which I taught you before. There's a graph, remember? Okay, so auxin at what level is toxic to the plant? No matter it is root or shoot. Is it high, very high, or very low? It's toxic to the plant. I mean, you don't have to give me the value. You don't have to give me the value. You just have to know the very high or very low. So it's very high. When auxin is in very high concentration, I don't care how much is it, okay? It is toxic to the plant. Last week, I've shown you a graph, okay? So what happened is that if you want the auxin to act as a weed killer, you have to use it at a high concentration. Understand? If you use the auxin at a very high concentration, okay, it can kill the weed because it can stop growth. No matter it's shoot, root, leaf, mati. Understood? Okay, that's something very important. And number 4, this one I have seen occasionally asked in exam. Okay, not as famous, but sometimes can ask in objective. They can use to store potatoes. Okay, auxin, one of the function can be used to store potatoes. All right. May I know, usually how do we know if the potatoes is rosak? If you teman your family members go to shopping lah, okay, how you choose the potatoes? How you know that this potato, I won't buy this potato? Okay, how you know that this potato is not good? They grow roots, 发芽. So 发芽 is not roots. They are not roots, you know. They are buds, buddings. You know, when they start to buddings. In English, we call this as buddings. Okay? In Bahasa Melayu, it's called betunas. They start to have the tunas to come out. So when they start to bud, you won't buy, isn't it? That means sometimes they say because of toxins and whatnot, isn't it? If you remember, auxin, okay, if you remember, auxin, is it more for apical dominance or is it more for lateral dominance? Can you remember? It's more for apical dominance or lateral dominance? Apical dominance. So if you came to my class last week, you will understand what's the meaning of the word apical dominance. That means auxin is mainly for the plant to grow upwards. not sideway. Sideway, you need the lateral buds. Who has the function to promote the growth of lateral buddings? Forgot already. Your cytokinin, isn't it? The hormone is called cytokinin. Only Sebastian remember. Okay, cytokinin. Okay, so that is the hormone for the lateral buddings. So auxin promotes apical dominance. So at the same time, they will prevent lateral buddings. So that is what causes the potatoes to spoil, the buddings. So auxin has the function to store the potatoes because they can stop the process of lateral buddings. Do you understand? Okay, so all these they are not explained in the textbook one. If you see the textbook, they just give you the points. They don't give you elaborations. So a lot of students, they cannot relate. How can they help? So actually, it has to do with the functions that we have learned last week. Okay, so these 4 functions of auxin, please remember forever. It's going to be very, very important. Even if you're a new student, you just joined the class today, you are a trial student, these questions will be definitely come out in your exam. I can be very, very sure. Based on my experience, it's very, very famous exam question. Okay, especially parthenocarpy. You have to remember this word, parthenocarpy. Okay, so this is auxin. So last week we have talked about ethylene, so I won't talk about that again. So we will just move on to talk about 2 more hormones. One is gibberellin. Okay, so gibberellin, usually in exam, what is it for? I hope you remember G. Last week I told you remember 2 words that starts with G. What are those? One is growth, pertumbuhan. So remember, gibberellin allows growth in all parts of the plant except which part? There's a part that gibberellin will inhibit also, just like auxin, the roots. If you remember, for the roots, gibberellin will inhibit. Okay, so one is G. So that's why can I use gibberellin to treat a dwarf plant? For example, I got a plant that is very short, not the root, I mean the stem. Okay, very short. So can I use gibberellin hormone to treat the plant so that it can grow taller? Yes. So it's once again talking about the function of gibberellin, isn't it? So I can use it, application, I can use it to treat dwarf plant. Okay, dwarf plant. Dwarf plants are basically short plants. All right. Okay, so that's one. Number 2 is germination. All right, germination. So percambahan. So I can use gibberellin, one of the functions, I can trigger germination of seeds. So if I want the seeds to germinate into new plants, I can use gibberellin as the stimulator. Okay, so growth gibberellin. But today, I want to introduce you the 3rd G in application, which is grapes. Grapes. Okay, the 3rd G, grapes. Ango. So if you see one application, they say what hormone will cause the grapes to be bigger and more juicy? Choose gibberellin. Okay, so gibberellin is also the hormone that triggers the formation of more bigger, juicier grapes. So this is something that you have to remember lah. All right, so this is the one for gibberellin. Last but not least, we have cytokinin. Okay, so I have already mentioned to you last week, cytokinin. You see the word cyto, it means cell. So you know that cytokinin actually stimulates cell division in all parts of the plant, including the roots. So roots stem leaves all also can be triggered by cytokinin. So it's like a growth hormone like that for the plant. Okay, so this is very, very important to trigger cell division, especially when we do a process called tissue culture. In Bahasa Melayu, culture tissue. That's what we studied in Form 4, Chapter 6. Okay, so this is to trigger cell division. So that's why one of the important application is in tissue culture. Talking about that, okay, in Form 4, in my Form 4 lesson, I am going to teach mitosis, meiosis, your cell division chapter in another 2 weeks. That means next week. All right. I know a lot of you, your cell division chapter sucks, especially students who didn't follow me last week. So your basics on that is not good. So again, if you would like to join the lesson, you just have to let Cindy know. Okay, so I told you before, for Form 5 joining Form 4, the cost is just 50 ringgit. So if you just want to join this chapter 6 lesson, it's okay, no problem. So I will add you in the group when we start chapter 6, and then you can exit the group after we finish off chapter 6 completely. If you want to continue with the lesson, then go ahead. But if you just want chapter 6, I can also tailor made for you. Why is chapter 6 so important? The cell division chapter. Last time, when before MCO time, I usually will do a seminar for Form 5 students to reteach the chapter, and usually a lot of students will join. But because now I'm quite busy, so I can't do it, I will just combine the Form 5 class with the Form 4. So if you want, you can join back the Form 4 one. So it's the same thing. Basically, I will teach everything. The idea is, if you suck in cell division chapter, you will suffer in a few chapters. All right. Number one, you will suffer in Form 4 chapter 15, which is reproduction chapter. So a lot of students, they don't know oogenesis. They don't understand about spermatogenesis. It's because their cell division sucks. So they don't know why suddenly diploid, why suddenly haploid. Because your cell division chapter is not good. That's one. Number two, later on, when we study, so some of your school teachers skip chapters, isn't it? Started with genetics, right? Some of your school teachers started with your chapter 11, 12, and 13, isn't it? So if you see those chapters, you notice that actually your cell division knowledge is very important. Without the proper knowledge of cell division, you will suffer when you study Mendel's first law, Mendel's second law. You will kick Yuan, definitely, because we involve cell division stages inside. We talk about anaphase, we talk about metaphase. That's why you have things like independent assortment and all sorts of things. So your knowledge on cell division must be good. That's the conclusion lah. All right. So in the earlier this year, there are some students who texted Cindy saying that when we reach chapter 6, please inform them. So now I inform you already lah, huh? Okay, I inform you already. I don't say I never inform you. You miss it already. That's it. Okay. So it's gonna start in another 2 weeks lah. Okay, next week or next week. I'm not sure because I'm now ending chapter 5 for Form 4. All right. So maybe next week, maybe next week. All right. So just to let you all know lah. Okay. Let Cindy know, okay, if you're interested. So this is the stuff. All right, the 4 things. Let us just move back to the notes. Let's see if I've opened the notes. Fill in the blanks. Okay. So aplikasi phytohormone. So we talk about the first one, auxin. So auxin menggalakkan pertumbuhan tanaman. All of us also know lah this one. Okay. So menggalakkan pertumbuhan akar adventitious. This is the adventitious roots that I said just now, just a spelling different. So it's basically pembiakan asexual. Okay. So what is the meaning of pembiakan asexual? Asexual reproduction means mitosis. All right. So there is no fertilization. It doesn't involve both parents. Like in this case, we use the stem, isn't it? So it grows into the leaves and all that. This is adventitious roots. Okay, so dia digunakan dalam keratan patang lah. Stem cutting method is a new growing method for the plant. Juga digunakan dalam parthenocarpy. Basically, parthenocarpy pengeluaran buah-buahan tanpa persenyawaan. Later you will understand in chapter 6. Basically, if you want to form fruits, you must fertilize. So if you want to form fruit without fertilization, then you must spray auxin lah. Okay. So pembentukan buah-buahan tanpa biji benih. It's just like getting pregnant without baby inside. It's very weird, isn't it? So sebagai racun rumpai, this is weed killer. We have to use the auxin at high concentration. Then you can kill the weeds. Melambatkan petunasan ubi kentang. So you can induce dormancy. That means you storage. It's for storage purpose of the potatoes. This is important because it can actually prevent the growth of the buds. So in this picture down here, you see seedless watermelon. Okay, this is also one of the techniques we use in parthenocarpy. But based on my experience, usually seedless watermelons, they don't taste as sweet as the one with seed. Am I right? Have you tasted this seedless watermelon before? Okay, so this page, if you are clear, you are done, you copied already, then you can write a letter 1 in the chat box. We move on to the next. Okay, remember, ladies and gentlemen, okay, please remember something, okay? The recordings for chapter the recordings for March will be deleted by tomorrow. Okay, so please take note of that. And for new students, I will add you in the Facebook group by tomorrow. Okay, so please be patient. I'll add you in tomorrow. Don't worry. So this one we have already explained last week, so I don't have to explain again. So gibberellin merangsangkan percambahan biji benih semasa suhu rendah ataupun keamanan cahaya yang rendah. So merawat tumbuhan yang kedil, this is dwarf plants, menghasilkan buah anggur, grapes yang lebih besar. Okay, cytokinin digunakan dalam teknik culture tissue, menggerakkan pembahagian dan pembesaan sel. So use it in the tissue culture techniques. Done? Okay. So this chapter, are you clear or not? Are you okay with this chapter? It's a pretty simple chapter actually. Okay, if you see, it's pretty straightforward. The exam questions also will be very straightforward. Are you okay? All right. So I think since we already finished the whole chapter already, why not we discuss the exercise first before we move on to chapter 6 lah? Okay. So just let us discuss the homework last week first, then only we move on to chapter 6. So let's talk about it. Okay. So let's look at the. Are you looking at the same page as mine? Yeah, you saw it. Okay, great. So let's look at Mayyen's answer. Okay, today we use Mayyen's answer as a reference. So Mayyen got 11 out of 12. That's very good. Okay, just wrong one question. So we see what we can improve. So this one is regarding last week's lesson, basically note 2. Note 2. So new students, you will not be able to access to this lah. So basically today we note 3, isn't it? It's note 3. So this is note 2. We just quickly go through it. So Johor Trial 2022, Raja menunjukkan pucuk tumbuhan yang tumbuh ke arah cahaya. Apakah yang menyebabkan tindak balas tersebut? So you have to see the question properly. Some students, they might be confused. They go and choose A. Pucuk memerlukan cahaya untuk photosynthesis. This is correct, but this is not the cause of the reaction. This is the reason for the reaction. So the best answer will be C. The cells shaded from light elongate faster. Sel-sel terlindung dari cahaya memanjang lebih cepat. If you remember last week, I told you before, if you look at the plant exposed from the light, auxine is a freaking shy person. They don't like to be at the spotlight. They like to hide. They are very shy. They hide from the shaded area. So the shaded area usually will have more than the bright area. So the answer will be C. Next is Negeri Sembilan Trial 2022. This question, a lot of students wrong. I already tell you last week already, you still wrong. Okay, so you see, they cut the coleoptile, so they put a block, but you must see carefully. The auxine is 100 mg per dm cube. High or low concentration, this one? 100 mg per dm cube. High or low? It's high. So you know that at high concentration of auxine, even the shoot doesn't elongate. So a lot of students go and choose C, but the key point is the concentration of auxine. 100 mg per dm cube. You refer back to the graph, it's inhibition. Okay, so it will not grow. Answer is donkey. This is a tricky question. Please remember this question. I can tell you in real exam, a lot of students will wrong this one. Okay, I have shown you this and I told you many times already, so please don't get mistake. Answer is donkey. So next is Pahang Trial 2022. So diagram shows an experiment studying effect of auxine on the response of 4 shoots to lights. So this is very common lah. Okay, so which of the following will bend towards the light? Okay, so P definitely will. This is agar block. I told you agar block allows diffusion of auxine, so it will also diffuse. So P and S. Answer will be C. Okay, because this one they put a black condom on it, so it doesn't get the light, so it will just grow upwards. This one no coleoptile means they got no auxine production, so they won't grow. So this is P and S. So next is Penang Trial 2022. Kepekanan oksin yang tinggi di bahagian bawah hujung pucuk merangsang perpanjangan sel. Bagaimanakah kepada oksin dalam sulur paut membolehkan sulur paut mengelilingi objek? Okay, so basically what it means is this question is not in your syllabus definitely, isn't it? But what causes tropism, no matter it's phototropism or geotropism, is always uneven distribution of oxygen. That means one side has to be more than the other. Okay, even though this is what we call stigmotropism, you don't study that in your syllabus in terms of the reaction, but there must be uneven concentration of oxygen. I know that for sure. Answer is A. So next is Terengganu 2022, also appeared in Terengganu 2023. So this was the graph that I mentioned earlier on. So you can see at 100 mg per dm3, even the shoot is in inhibition, okay? So they tell you part A and part B. You know part A is root, part B is shoot, okay? So now they're asking you, the concentration is 10 to the power of -1. That means at this point, okay? So you know part B definitely is promote elongation, but part A is already inhibition. So part A inhibit, part B stimulate. So the answer will be C. Next question, SPP trial 2023. So diagram shows the germinated seed kept in a dark cupboard after 3 days. Okay, so it doesn't matter because this one is geotropism, so I don't care about the light. What causes the response shown by X and Y? So you can see this is the plumule. You see it goes upwards. This is the radical. It goes downwards. So the reason obviously is because there is more oxygen accumulated at the bottom. Answer will be C, because this is geotropism. That means the gravity pulls the oxygen downwards, like what I explained to you last week clearly. Next is Kedah trial 2022. So we have a light source coming in from a hole in a box. So they ask you, which of the following statement explains the situation? So I have already explained this just now. Basically, at the shaded area, which is S, they will elongate faster. So the answer will be A. So next question. So this is pretty simple. If the light comes in this way, how will the shoot grow? Basically, it will grow towards the light lah. So the answer will be C. Pretty straightforward lah, this one. Okay, Kelantan trial 2023. So diagram shows the tropism response in the coleoptile. So I got 4 reactions over here. So this is agar block, black cloth, blah blah. So which of the following shows the response of the shoot? Okay, so you know that this one definitely will bend towards the light. This one will not, because there's a black cloth over it. They cannot get the light, so it will grow upwards. This one is agar block. Agar block allows diffusion, also will grow towards the light. So this one, coleoptile, is cut, so it will not grow. So you see which one follows it lah. So bend upwards. So the best answer will be A, isn't it? So A. This one will grow upwards lah. Oh, okay. It wouldn't be the same as this ma. Correct not? Because it's black cloth ma. The oxygen is still there, just that it's an even distribution. It go upwards. Answer is A lah. Okay, Kedah trial 2023. Which of the following statements explain the situation in diagram 17? So you see that in this picture, this is the plumule, it goes upwards. So again, we talk about the concentration, high concentration of oxygen at S. The lower part, okay, stimulates the elongation of cells at S. So answer is A. Okay. All this we have explained last week already in the class. So once again, light, agar block, coleoptile, it will bend toward the light. Okay. So don't. The key point is, if you see agar block, it doesn't change anything lah. Okay. But if you see mica block, then it blocks the diffusion. So for this, the answer will be C. MRSM trial 2024. Diagram shows phototropism. So once again, they put an opaque cover. So they will definitely grow upwards lah. Which one is it? Answer is boy lah. Okay, that's all. 12 questions. So I can be very, very sure. Okay, your friends would simply do the questions. Okay, for those who never studied properly, they will wrong a lot one, because there are a lot of tricky questions here. But for those students who followed my class last week, okay, you will perform well lah. So you only wrong one question. I think it's a careless lah just now for that. Okay, so let's have a look at the summary. Oops. Okay, So I still did. This is not a good performance, lah, to be really honest. Okay, if you ask me, this is not a good performance because even though a lot of students actually got correct, but I want the graph to be pushed towards this side. You saw last week's graph, right? This is not very good. Okay, this is not good. All right, you must be over this side. So this distribution is not very good, lah. That means there are some questions that you all confused already. So like I said, I'm always available. If you have any questions, you can always ask me. Okay, so you have to aim to be over this side. Right? Then we just look at the structured questions together. Jom. Boom. Oh, okay. So I gave you a few questions. So let's do it together. It's going to be quite easy, the questions. This came up twice, once in Kedah, once in MRSM, 2022 and 2021. So, nyatakan dua ciri-ciri gerak balas yang ditunjukkan dalam Raja. So basically, they are trying to ask you about tropism. This is asking you about tropism. So you just have to tell her, tropism is slow, not apparent. Response is permanent, depends on direction of stimulus. Any 2 out of 3 points, you get full mark. This is something that's very basic already. I wouldn't repeat it again, okay? So based on the diagram, state the role of auxin in the response. Basically, they ask you to talk about phototropism, positive phototropism. Am I right? So once again, only 3 marks. So you should talk about what's the function of the auxin, compare the concentration of auxin. So it's always higher in the shaded region. Okay. Talk about the elongation rate, which side elongate faster. And last but not least, the direction of the root bending and what is the name of the response. So this is basically, you can get it directly from my notes itself. It's directly over there. So you should be able to do the questions easily. Okay. So once again, you don't have to copy the exercises lah. Okay. All these you can access from today's recordings, and later on you can always watch back the recordings and copy the answers back to your notes. Most importantly, in this 1.5 hours, I want to give you full input. Okay. Full impactful input. You just learn the way to answer. So this is another question. You can see, basically they put the manga inside the bekas nasi. This is like last time what the old people tend to do, isn't it? They want the fruit to ripen faster. They put it inside all these rice containers. They wrap it up. So that's the reason is, I told you before, there is a hormone called ethylene. So this hormone is ethylene. Okay. So the function of the ethylene is basically to speed up the process of the ripening process. Okay, and the special thing of ethylene is, so when they ask you why you place it in the rice container, because ethylene is a gas hormone. Tell first, ethylene is gas. So they can be trapped in the rice container and they can stimulate the ripening of the mango. So once again, what is the meaning of ripening? It has 2 meanings. One is the fruit becomes softer. That means the ethylene can stimulate the production of cellulase to break down the cellulose in the fruit cell wall. That's one. Number 2, the fruit will be sweeter because the acids and the complex carbohydrates in the fruit will also be broken down into simpler sugar like fructose. So that's important. 2 things. One is the texture, which is softer. One is the taste, which is sweeter. That is what defines the ripening process. Okay? Which I have explained last week as well at the end of the lesson. So this is a Terengganu trial 2022. So a fruit seller found that a pile of fruits that he sells quickly spoil, so that it cannot be sold, and the seller is at loss. Based on the biology knowledge, suggests a way that can be used by the seller to prevent the fruits from spoiling. So why the fruits spoil is basically because of the ripening of the fruits. So there are 2 ways you can do it. One, like what I told you last week, you put the fruits in the refrigerator, because usually they can slow down the process of ripening as they delay the release, or they destroy the release of the ethylene gas. That's one. 2, you can also separate the fruits, especially the ripened fruits with the unripened fruits, because the more ripened the fruit, the more ethylene will be produced. So if you mix all the fruit together, definitely they will, the ethylene gas is flying everywhere and the fruits will be ripened up much faster. So this is the two suggestions I can give you. You can choose either one. Okay. So this is Malacca Trial 2021. So I just show you this is a type of question they ask you in exam. So basically they're asking you, so you see the question carefully. They just want you to talk about radical. Radical is the roots. So why they grow towards direction Y. So this is also what we have explained last week. You always talk about the oxy, talk about the stimulus, talk about the comparing comparing the concentration, talk about the direction of the rate of elongation, direction of bending. It's the same. Okay. In conclusion, this is the same as our notes. Nothing special. So I just want to show you that whatever I show you previously are real exam question. It's memang like that one. Paratrial 2020. Okay. So it's an experiment to study the effect of hormone on the growth of a maize coleoptile. So you can see they go and put the aluminum cover over the coleoptile. They put a light source over here. So you know that after 1 week, this is something that we studied just now in the objective questions as well, isn't it? So you know that the maize will grow upwards. Okay. Why? You need to mention the key point, even distribution of auxin. Because they don't get the light due to the opaque cover, the aluminum cover. So the auxin distribution is even, so they will elongate evenly lah. So you know that which are the keywords, isn't it? Okay. So this is a Terengganu Trial 2023. So we talk about this graph already just now. So they ask you to give a conclusion based on the graph. So I know that based on the graph, this one is a bit different from the usual graph. Based on the graph they give you lah. Okay, don't use your knowledge. You have to base on whatever they give you. So I know that for roots, the maximum elongation is at 10 to the power of -5. For shoot, the maximum elongation is at around 10 to the power of -2. This point, right? So what it means is that if I talk about 10 to the power of -2, okay, actually the auxin will promote elongation of the shoot and inhibit the elongation of the roots. That's the conclusion. That's the usual physiological dose of auxin in the shoot and root, which causes phototropism and geotropism. Any questions that you all want to ask me? When I discuss exercise, I will go quite fast because I think mostly you need to know the concept only. You really don't need to, word by word follow it. That's not the point to study lah. Okay, you have to know how to answer instead of what is the answer. You understand what I mean? Okay, how is always more important than what, because if you know the how, they change the question, you can still answer. But if you memorize answer scheme, in which there's some tuition centers, some teachers who promote you all to read answer scheme, then it makes you not thinking. You understand what I mean? So most importantly, when we talk about answering biology questions or any subject, is the critical thinking behind it, not to become a robot to answer questions just based on the schemes. You have to think how to answer. Okay? That's why you notice how come students can be very performing in Form 4, Form 5, SPM level, straight A+. Once they get up to higher level, for example, they enter the medical course. I ask you, in government universities, for example, UPM, UKM, for example, you think the students straight A+ or not? All of us who enter into government uni, all also straight A1 Why medical school still so high failing rate? Why so many people still fail one? Because the way a lot of the students study, they still use the Form 4, Form 5 way of memorizing to study. So I'm teaching you all to study biology in the very beginning onwards. The way you think must be critical. You must understand the reason behind it and not to just follow like that. No, you must think the reason. So that's why when I teach also, I try to make you understand why is it like that, instead of, just tell you the point. Everyone can read correct or not? You got eyes, you got the eyes, you got textbook, you can read already. So if I read the textbook for you, or if I just read out the notes for you, then there's no purpose for my existence, isn't it? And my point, my part is to tell you what I understand to make your life easier. So you need to follow my course properly. Like last week, 60-something students answered the objective questions. This week, drop already 55 only. So next week, if drop some more below 50, you know our promise, right? What will happen if the answering rate below 50 students? Okay, so please, today is already 55. It's already quite dangerous already. It's almost there already. Okay, so next week, do better. All right, so can we move on to chapter 6? If can, write okay in the chat box and move on to the next chapter. Okay? Any questions that you all have? Any questions that you all want to ask me? Anything that you all don't understand that you want to clarify? Please don't be shy. Just ask me anything. Okay? Any questions? Okay, so all of you already say okay. That means I can move on to chapter 6. Let's go on. Boom. Okay, so this one just how we have already explained. Pop. Okay. So chapter 6. So chapter 6 basically is talking about pembiakan, reproduction of angiosperms. Okay, reproduction of angiosperms. So first of all, what is the meaning of angiosperms? Angiosperm is not the sperms of the angel. No, angiosperms in a simpler word means flowering plants. Flowering plants. Tumbuhan berbunga. So whatever plants that can produce flowers, they belong to angiosperms. So in the hierarchy of the plant, okay, so for example, in the hierarchy of animal, who is the most advanced one? In the animal kingdom, who do you think is at the top of the animal kingdom? That means really is the best animals lah. Who do you think it is? In the animal kingdom, who? Lion, monkey, human lah. Of course, the answer is human lah. You don't think human is an animal? Under animal kingdom, human is Homo sapiens. We are under Animalia kingdom. So don't think that humans is not animal. Human is what is human? A fungus? No, right? So humans are animals. We belong to the apex. Okay? So in the plant kingdom, who is at the apex? Flowering plants. This is the most advanced plants. Because when we talk about reproduction of the plants, two types of reproduction we have. One type is asexual reproduction. Okay, that means some plants, they don't use flower to reproduce. They reproduce by producing spores. They reproduce by producing cones. They don't use flowers. So those plants are a bit lower ranking than flowering plants. They're more advanced in terms of evolution. So the type of reproduction they do is called sexual reproduction. So it's called pembiakan sex. So in sexual reproduction, the most important thing is what? What's the main difference between sexual and asexual reproduction, guys? You should be able to tell me at this point of Form 5. What's the main difference between pembiakan sex and pembiakan asex? The main difference lies in the word start with F, which is fertilization. Some people say F word. The F word here is fertilization, right? So, asexual, no fertilization, but sexual, we have fertilization. So, when we talk about fertilization or persenyawaan in Bahasa Melayu, you must have gametes. You must have gametes. Gametes are the cell sacs, like sperma ataupun our cell telur lah. Okay, so gametes must be produced through the process of meiosis. So that's why later on we are going to study how do we form the gametes. There must be male gametes and female gametes, and how they do fertilization, and how the whole process goes. So today, let's see how far we can move on. Okay, so maybe today, if you are lucky, if we are quick enough, then maybe we can talk until the formation of embryo sac. That is something that is pretty important in your syllabus. Hope we can reach that. Okay. So without further ado, let's just look at the structure of the sexual organ. Have you ever wondered, okay, why do we give flowers during Valentine's Day? Why? Actually, I mean, romantic. Okay, romantic. Okay, all right. Flower. What is flower in plants? Actually, what is the role of flower in the plants? What organ is it? Yeah, it's an organ, of course. It's an organ. What organ is it? It's a sex organ. Sex organ. So, to be really honest, as a biology point of view, I only gave flowers to my wife once during proposal. Okay. After that, I never give. And then people always tell me, Stephen, you are really not romantic. And I'll tell them why. They say I never give flower to my wife. I say, why you want to give a bouquet of dicks to my wife? Isn't it? So if you give like 100 roses, to me, it's just like 100 dicks hanging around. Okay? They are all just sex organs. Isn't it? So for what you want to give a bunch of sex organs to your wife where she only need one? I mean, that's my point lah. Correct enough. So I don't give flowers to my wife, all right? Because to me, it's just a bunch of sex organs. In the plants, flowers are sex organs, and you need to see there are two types of flowers in this world. One is called unisexual flower. Unisexual flower is just like you and I, hopefully, that I am a male, you are a female, you have female sexual organ, I have male sexual organ. So you don't see me having male sexual organ and have vagina at the same time, right? So that is both sexual organs. So we are all unisex. So I have male sexual organ, you have female sexual organ. So flower also is the same. Some flower are unisex. That means they only got the male part and they only got the female part. But mostly in our syllabus, we talk about bisexual flower. That means they have both the male and the female sexual organs in one flower. So let me show you. Okay. Okay, my drawing sucks and I know it, but it's enough for you to understand lah, basically. So, the side one, I'll show you what is it. This one, the side one, okay, which is from here to here. This one that I label, okay, which is also this thing, the green color one that I label, okay. This is the man part of the flower, the male part, okay, the male part of the flower, this whole thing, okay. They scared you don't know. So that's why this is called stamen. For students who don't know this is male part, I really want to slap you because they will tell you they are men. Right? So stamen is the male part of the flower. All right? So it's made of two parts. One is this, which is called as anther. In Bahasa Melayu, just remove the H. Okay? This is H, by the way. Yes. My words, my writing. No wonder the pharmacists always call me, say they cannot understand my words. So anther, okay? So anther, there's a H over here. In Bahasa Melayu, there's no H. All right? So this anther is like the testis of male. All right? So testis for the male produce sperm, isn't it? So what is the male sexual gamete for the flower? What is the male gamete for the flower? Anyone knows? Anther is the part that produces the gamete. This is not the gamete. Anther is the part they produce the gamete. What is the gamete? Seed. Seed is not. See, you're all confused already. Seed is later one. After fertilization, only you got seed. It's called pollen grain. 花粉 in Chinese, we call 花粉. Pollen grains. Heard of it before? Okay. Right. Pollen grains. In Bahasa Melayu, it's called debunga. 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 Okay? Flower semen. And then I'll tell you what the flower what the bees usually go to take. Okay? Pollen grain. So this is basically the male gamete. Alright? For the flower. So you can see that this anther, basically they are connected to the ovary. This is the ovary by a stalk. There's actually something like a stalk that connects the ovary to the anther. So this stalk is called filament. Okay, this stalk is called filament. This part is called filament. The only function is for support lah. They support the anther. That's all. So I repeat, the male portion of the flower is called stamen. They had two parts. Anther, the top part is used to produce the pollen grain, which is the male gamete. The below part is a stalk called filament, which is used to support the anther to the ovary. Okay? So now we look at the female part. The female part is so dominant as usual, so they will be in the center. This center part is the female part. This one. This one is the female portion of the female portion of the flower. So it is known as carpel. Carpel. Okay, maybe I erase this one so that you don't need to see. It's the same thing basically. So one flower can have many and can have many stamens. I just draw two, but in reality they can be many. All right. So carpels. All right. Last time, I don't know whether you all got see before. No? There's a type of flowers that usually present in the primary school. Red color one. And then last time as a kid, I like to pluck it and just go and suck it. I don't know whether you all got do it before. No? Anyone have done it? Or it's like our time? Usually in primary school. I don't know why. You never see it before in secondary school already. So yeah, it tastes sweet, right? Do you know what do you know what you are sucking? Any idea? All right. So never mind. So let me just give you elaborations. This is the one actually, this stick over here. Okay. So what is this thing over here? This is called stigma. This part. This part. All right. This part is called stigma. All right. So stigma, I want you to remember a few important points. Number one, it starts with the letter S. So I want you to remember there are two things you have to remember start with letter S. Number one, stigma is very sticky. Stigma is very sticky. Number one. So that means later something must stick on it. What stick on it? Must be the pollen grains lah. Okay, so the pollen grains later will come and stick on this. So it must be very sticky. And on top of the stigma, they must have some sugary solution. That last time when you taste, it is a bit sweet. So what sugary solution is that? That is sucrose, which is another S. So stigma, S, think of stickiness, think of sucrose. Later you will know what's the function of this sucrose. It's not for you to suck. It has a function for the flower. Okay? So this is the first part, the stigma. Number two, down here, we have something called the style. This one. Okay? So this part is called the style. In Bahasa Melayu, it's also called style, just it becomes S-T-I-L, style. So style basically is just like a filament, but the function is to connect the stigma to the ovary. So this part over here is the ovary. Okay? So let me enlarge the ovary and show you what is inside the ovary. So I zoom in the ovary. This is pretty important. I think you need to know about this. Let's zoom in. Okay? So, inside the ovary, okay? So this is ovary, ovary. Inside the ovary, there is something called ovule. Ovule. Draw second layer. In your syllabus, they usually will draw the ovule very big. Okay? But not necessarily they are so big. It depends on the type of fruit. Because, okay, I tell you, if you remember what I told you just now, fruit comes from which part of the flower? Fruit. For example, you eat the mango, right? You eat the mango. The fruit comes from where? Which part of the flower? is from the ovary. Just like I told you, right? It's from the ovary. So that means ovary later becomes the fruit after fertilization. This is the fruit of the flower of the plant. This becomes the fruit. So this ovule is inside the ovary, right? So in the fruit, what do you have inside? Basic, simple question. What you have inside the fruit? You go and makan the fruit and tell me what you have inside the fruit. Papaya, mango, what do you have? Seeds, right? So if ovary becomes the fruit, this ovule becomes what later? The seed. Ovule is the seed later. Ovule is the seed later. So to make your life simple, usually your exam, they will draw only one ovule. But you need to remember this. You have to remember this. Okay? Very important point. It depends on the type of fruit. For example, if today we talk about mango, how many seeds the mango has? How many seeds the mango has? Only one, right? So this is the fruit like mango. But if today I draw fruits, for example, I talk about fruits like, let's say, we talk about fruits like papaya. Do you think they only got one ovule in the ovary? Or they got actually many? Many or one? Many lah. So sometimes in the exam, there can be a tricky one, They can draw something like this, and then inside, they draw like this. They draw like this. So student confused already, because in their simple, innocent mindset, ovule always look like this one. They never notice, this one is the ovule. Because this is the fruit that has 6 seeds, for example. Do you understand what I'm talking about? You know what I'm talking about or not? Okay? So, to make your life simple, I will only use one ovule to show you what happened. So this is the fruit, like for example, mango. Alright? But in reality, there are some fruits that have many seeds, which means in the ovary, they got many ovules one. Understand? Ovule becomes the seed later. Ovary becomes the fruit later. Understand? But is the seed the embryo? Is seed embryo? Or seed contains embryo? Which one is correct? Seed contains embryo or the seed is embryo? The seed contains embryo. So that means inside the ovule, you still have something. You still have something inside. This layer is called embryo sac. In bahasa Melayu, it's called pundi embryo. Pundi embryo. Okay? I pronounce the word very properly. Pundi embryo. So this is the one, okay, that you have inside the ovule. So, in simple way, I just want my students to remember ovary. You remember 3 layers at this point. As a new student, you remember 3 things. Okay? Ovary, ovule, embryo sac. If you don't know what is it, I will show you later what is embryo sac. Okay? So let me just give you a quick summary. The female part of the flower is called the carpel. It has 3 parts. Stigma, which is very sticky, contains sucrose. Style, which connects the stigma to the ovary. So in the ovary, we have ovule. In the ovule, we have something called embryo sac. Later, then some students ask me, okay, sir, then what is the female gamete? If the male gamete is pollen grain, where is the female gamete? You notice that in the embryo sac later, okay, there will be one cell in the middle here. And that cell is called egg cell. In bahasa Melayu, we call this a cell telur. So this is the female gamete of the flower. So the female gamete is called egg cell. The male gamete is called pollen grain. All right. I think I haven't put this set of notes in my computer. Let me just put it back. Give me a moment. This is note 1. Why is it loading so long? Okay. So can we fill in the blanks? Okay, so Form 5 Chapter 6 Note 1, I hope you all have already downloaded the notes already. It's in the Google Classroom. So, I repeat one time in Bahasa Melayu. Struktur bunga, alright? So, bunga dia merupakan organ pembiakan angiosperma. Angiosperma is flowering plants, bunga, tumbuhan berbunga lah. So, they have two parts. Stamen dia adalah organ pembiakan jantan, jantan, alright? So, this is this thing, this one, this is all stamen. So, dia ada dua bahagian. Satu adalah anther, which menghasilkan debunga. Debunga is pollen grains. Okay? Debunga. So, filament is this yellow color stick they used to menyokong the anther. Carpel basically is the organ pembiakan betina, which is this middle one. So, dia ada tiga bahagian. One is called ovary, which is this part over here. So, ovary dia mengandungi ovule. Sorry in Bahasa Melayu, ovule shouldn't have E Sometimes they go and auto-correct my thing. Ovule. So you can see inside here, there's something called ovule inside. Okay? So dia menghasilkan pundi embryo. What is this? Later I'll show you. So stigma. So stigma, dia adalah struktur di mana debunga melekat. So remember, stigma is very sticky and they have sucrosa on it. Style is the one that menggabungkan stigma ke ovary. Yes. Another name for carpel is pistil. You're right. Pistil and carpel you can use interchangeably. Okay. This one. Oh, in English there is pistil. Pistil and carpel are the same. Okay, so let's talk about something kebat lah. What's the function of this petal? Petal. Petal. What's the function of petal? This one. Protection. Really? It really protects the flower? The function is to attract the pollinators. You know pollinators? The one who come and take the pollen grains. Because when the flower is attractive, sometimes it can be very colorful, isn't it? Attractive, number one. Number two, they can release a very nice scent. They smell good. So the insects or the small animals like birds, they will fly over there. So they kena the pollen grains on their legs, and then they can actually fly to the stigma of another flower. Okay, and they do the process of pollination. Pollination and fertilization, are they the same? Pollination and fertilization, are they the same? What do you think? Guess lah. If you don't know, also guess lah. Are they the same? Pollination and fertilization is different. Pollination is what? In Chinese, how do we call it? Pollination. 播种. So pollination means when the pollen grain from the anther, you transfer to the stigma. That is called pollination. Pollination doesn't mean fertilization. Fertilization is when the male gamete nucleus fertilizes with the female gamete nucleus, forming a diploid zygote. Okay, so fertilization happens after pollination. You must do pollination first. That means the pollen grain must go from the male anther to the female stigma first. And this petal has a very important attractive characteristic that menarik perhatian serangga ataupun binatang kecil lah, like burung. What is sepal then? What is this sepal? Sepal has not much of function. Basically, before the flower blossoms, that comes from a bud. So this sepal opens up and then the flower will be blossom. Sepal only has some supportive functions to support the flower. That's all. Okay? This is going to be a 5-star exam question. Very important exam question, and I can be very sure some of your schools will come out this in your essay. Essay, this is essay, not structured, essay question. So usually the question will sound like this. Can you please compare the male and female reproductive organs in the flower. So comparison must have similarities. So similarities, kedua-duanya menghasilkan gamete. Both produce gametes. One is male gamete, one is female gamete, but both are gametes. Two, kedua-duanya dalam bunga. They are all in flowers. So perbezaan, alright, bahagian jantan, we have to call the name. Stamen lah. So what it contains, anther and filament lah. What they produce, the bunga lah. Where is the location? They mengunjung keluar dari dasar ovary. That means they come out from the side, from the base of the ovary. So for the bahagian betina, very simple. It's called carpel. It has three parts: stigma, style, ovary. It produces pundi embryo, which I will explain to you in a while, and it's at the middle of the flower. Can you answer this question in an exam? If I give you a 4 marks essay on this, do you think you can do it? Now you just baru study, of course, memory very fresh. But in the real exam, if I give you this a 4 marks essay, can you do? If you cannot do, you just draw back the flower that I showed you just now. Right now. Stamen, carpel. Stamen got anther, filament. Carpel got stigma, style, ovary. Anther produce pollen grain. Style, the stamen has pollen grain, and the carpel, they have embryo sac. One is in the middle, the dominant ladies. The males are all at the side, coming out from the base of the ovary. You don't understand this last point? This last point. Okay, fine. See this. Do you see the stamen? They all come out from the base of the ovary. That means they come from the ovary lah. Whereas for carpel, they are in the middle of the flower. Even if you see if you see the English version, also is the same. You can see the carpel, pistil is carpel lah. The carpel, they're in the middle, and then you can see the stamen all comes from the base of the ovary. This is the meaning. Do you understand? This is ovary correct or not? Then they come from the base of the ovary Any more questions you all want to ask me regarding this? If you're clear, please write the word 2 in the chat box. I can move on to the next part. If you're clear lah. Okay, great. A lot of you already understood. So we move on to the next part. Jom. In human male, in human male, we call the process of producing sperm as spermatogenesis. Right? We studied in Form 4, Chapter 15. In plants, they don't have a special name for it, but we also have to study. So the next thing that we're going to learn is called the formation of pollen grains. So how do we produce the bunga? So first of all, I need to show you an anther. So let's say this is an anther lah. Alright, this is the filament. So this anther, alright, if I want to show you how it looks inside, you will see actually it looks like this. My drawing is very bad lah, but it's something like this. So it has this place called as the pollen sac. Pollen sac. Pundi de bunga. Pollen sac. So each of the anther has 4 pollen sac. One anther has 4 pollen sac. So inside this pollen sac, okay, we have a lot of diploid cells known as, okay, do you still remember what do we need when we talk about formation of gametes? If you follow my class in Form 4, Chapter 15, I told you, you have 3 requirements. Number 1, you must know the name of the cell. Number 2, you must know whether the cell is diploid or haploid. Number 3, you must know what process the cell undergoes. So these three things must have in your structured or essay questions. You lack one also cannot. So if you remember in spermatogenesis or in oogenesis of humans, the first cell that start the process is called the germinal cell, right? The germinal epithelial cells. But for the flowers, it's much easier. The name is called, I'll explain to you, microspore mother cell. What is microspore? I will show you in a while. Why mother? Because they are the first cell. It's like a mother give birth to the children like that. So we call it as a mother cell. Okay? In Bahasa Melayu, not so sexist one. In English, you notice they're very sexist one. They always call daughter cell, mother cell, right? Always talk about the ladies, all right? Macam father no use like that. Always mother cell, daughter cell. In Bahasa Melayu, usually they don't have this. They just call it cell anak or cell induk. This is called cell induk microspora. Okay? Basically, it's the first cell that undergoes the process. So in the formation of pollen grains, you straight away start with the process of meiosis. So the process they do is meiosis. Because you want to form gametes, right? So you must do meiosis lah. So the microspore mother cell that initially was diploid, after they undergo meiosis, they will form microspore. Okay? So now you know why is it called microspore mother cell? Because they produce microspore. So if we got microspore, what is the meaning of the word micro? What's the meaning of the word micro? Small. Small, right? So if we got microspore, do you think we got megaspore? Megaspore. Do you think we got megaspore? If there is nothing big, where got small eh? So if I tell you this is microspore, that must be megaspore. So who is the megaspore? Megaspore is the female part that happens in the carpel. Now we are in the stamen. This is in the anther, right? Okay? So males, we are the very humble one. We call ourselves microspore. Later when we study the female pundi embryo, formation of embryocyte, you will see where is the megaspore. The female is there, the megaspore. For male part, microspore. How many microspores are produced from one microspore mother cell after undergoing meiosis? Basic question. So one meiosis produces how many daughter cells? How many? 4 lah. Correct or not? 4. So you get 4 daughter cells. And what is the chromosomal number? Diploid or haploid for the microspores? After undergoing meiosis, definitely it will be haploid. So easy, right? But there's a plot twist here. So let's say this is the microspore. Okay. So they undergo meiosis, so you produce 4 cells, right? Sometimes we can call this as a tetrad also, but usually we call it as microspores lah. Okay. So the plot twist is, if we zoom into each of these microspores, okay, so this is initial the microspore mother cell 2n. Now there are n already, isn't it? Microspore. Okay. What happened is the nucleus of the microspore will undergo mitosis 1 time. That means, this is something very important. The nucleus of the microspore will undergo mitosis 1 time. Which means now they produce something that have 2 nuclei. Each of the nucleus is haploid lah, because they use mitosis lah. Let me ask you a K1 question. Besides pollen grain, what is another thing that you know has 2 nuclei one? Just a very simple question. Anything that you know has 2 nuclei besides pollen grain? No one know. You all really are. Paramecium lah. How many nucleus paramecium has? 2 lah. one macronucleus, one micronucleus, remember? So now I want to add on one more thing in your syllabus that has two nuclei. Pollen grains, the bunga. So pollen grains also got two nuclei. You have to remember that. Okay? So one nucleus is called tube nucleus. So in Bahasa Melayu, it's called nucleus tube lah. Okay? So tube nucleus. Tube nucleus. Another one is called generative nucleus. Nucleus penjana in Bahasa Melayu. Okay? There is a reason why they call like that, which I will only explain to you next week. Tube nucleus because they go and form a tube. Generative nucleus because they generate something. So we got two. So once they grow these two nucleus, we call this as a pollen grain. This is a pollen grain. The bunga. So let me repeat this simple essay. Microspore mother cell with its diploid undergoes meiosis to form 4 haploid microspores. Each microspore's nucleus undergoes mitosis for one time to produce 2 nuclei, namely tube nucleus and generative nucleus. Then a pollen grain is formed. Let me show you the notes to make your understanding even better. Okay? So, debunga dihasilkan di dalam enter yang mempunyai 4 pundi debunga. So, if you look at the enter, you can see there are 4 pollen sacs inside. So, setiap pundi debunga mengandungi banyak sel induk microspora yang diploid. So, this is the first one, sel induk microspora. So, setiap sel induk microspora membahagi secara meiosis dan membentuk 4 sel microspora yang haploid. So this is your cell induk microspora. When they undergo meiosis, they can form 4 microspora yang haploid, isn't it? So we can call this as a tetrad lah. Okay? So nucleus dalam setiap sel microspora akan membahagi secara mitosis untuk menghasilkan nucleus penjana dan nucleus tube. You can see nucleus penjana, nucleus tube. Microspora then berkembang menjadi debunga yang matang. So there are only 2 processes that happen. One meiosis, one mitosis. That's all. Any questions or is there any difficulties that you don't understand? Are you okay? Understand? So this is something very easy. If you are clear, you can just write letter 3 in the chat box. I'll move on to the next part, the last part. So we still have about 6-7 minutes. Okay, can understand? This is something pretty easy, I guess. Okay, so we move on. All right. This is the part that students always confuse. This is the part that students always confuse. I don't understand why you're confused. You always confuse. Embryo sac. So in bahasa Melayu, pundi embryo. Okay? So you must first understand where is it first, where it happens. So in the very beginning, I just show you the top part. This is the style. Okay? So down here, we got the ovary, right? So this is the ovary. All right? So inside here, we got something else. What do we call this? What do we call this, guys? This is called ovule. 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 All right? So in the very beginning, there is no embryo sac. So what we have in the beginning, inside got the cell. So I want you to make a smart guess. Just now when we form pollen grain, what is the first cell that undergoes division? What's the name of the cell? Repeat again in the chat box. What's the name of the cell that undergoes the first division process just now for the pollen grain? Microspore mother cell, right? Microspore mother cell. Make a guess. Just now I tell you already. Male is the humble one, microspore. So female one, you guess what's the name? Megaspore. So the first cell is called megaspore mother cell. So megaspore, megaspore, mother cell. Diploid or haploid, this cell, 2n or n? Must be 2n. Next question, what process this cell happens? Same lah, like just now, what process they do? What do you think the process they will do? Meiosis, right? Definitely it's meiosis lah. Because you want to produce gametes, right? So you produce meiosis already. You produce 4 name of the cell. Same like just now. Just like it's microspore, right? This one produce what? Megaspores. Diploid, haploid? Haploid. Okay, fine. Correct. So have a look over here. So can I say, in the very, very beginning, this is the megaspore mother cell diploid, right? So they undergo meiosis. So they are going to produce 4 cells like this. Am I right? They're going to produce 4 cells like this. Understand? Now this is the tricky part. I ask you a quite simple question. Every month, right? Female releases how many secondary oocyte? Or in simple word, how many secondary oocyte is ovulated? 1. But if we do meiosis, supposedly we should produce 4 gametes. Why only 1 is ovulated? Can anyone answer me this question? For female? Store? No. Why only 1? Every month you ovulated for female. Why not 4? Because the rest are degenerated as polar bodies. Remember the Jasad Berkutub, your what do you call that? Polar bodies. Forget already? They degenerated as polar bodies. So the same case over here. Out of the 4 megaspores, 3 of them will degenerate. That means out of the 4 megaspores, 3 will degenerate. Left only 1, which looks like this. Then students ask, in the beginning also like this. Now also like this. No. In the beginning, it was diploid. Now it is haploid. We got the same, even though the number is the same, but the content of the chromosome is different. This cell, do you think it deserves the name as megaspore? Do you think this size looks like a megaspore? No. So what it has to do, the remaining one, they will start to grow. So when they grow, they become like this. So this is the megaspore. No, it's not an egg cell. Megaspore. If you remember just now, the pollen grain, does the story end here? Or there's something that happened after the meiosis process? What happens? Can anyone tell me in the pollen grain formation? What happened? The nucleus divide how many times? How many times the nucleus divide of the microspore? 1 time. Divide by what process? Mitosis or meiosis? Mitosis. So over here, same thing happened. But remember, the nucleus, because this is female, everything need to be big, everything need to be more. So the nucleus divides by mitosis how many times? 3 times. Epic or not? So if you undergo mitosis 3 times, how many nuclei are you going to produce? How many nuclei are you going to produce? 6. You must come to my class, Devesh. I mean, you are already in my class, but you don't just use 2 times 3. Okay? It's 2 to the power of n. Understand or not? N is the number of division. So if you undergo division 3 times, then it's 2 to the power of 3, which is 8. You produce 8 nuclei if you do mitosis 3 times, not 6. You don't just times like that. It's a power. It's exponential increment one. Okay? So, your job now is to know how these 8 nuclei going to arrange. Some of them will even start to form cells. Let me show you. This is very important. Let me delete this. This is a megaspore. Now we've got 8 nuclei, right? Okay. 2 will remain in the middle. So 2 will be in the middle. So these 2, we call them as the polar nuclei. I repeat, polar nuclei. In Bahasa Melayu, we call them as nucleus berkutub. Okay? Polar nuclei. Okay? Polar nuclei. 3 of them will move to the opposite pole, which is over here. Okay, why I call this as opposite pole? Because usually in the ovule, right? In the ovule, there will be a very small opening over here. Let's say over here. There is a very small opening over here. This green point. This small opening is called micropyle. In English, it's called micropyle. M-I-K-R-O-P-I-L. Okay? It's direct translation. Micropyle basically is a small hole in the ovule. Alright? So the micropyle is over here. This is like the vagina of lady like that. The lubang. Because you must go in later. Something need to go in. I'll show you next week. Okay? So these 3 cells that is opposite the lubang is called antipodal cells. I repeat. This is called antipodal cells. Why they call as antipodal cells? There is a reason. What is the meaning of the word anti? Anti means opposite. Right? Opposite. Podal comes from the word pole. So antipodal cell means they are in the opposite poles. So the nuclei, they move to this side and they already differentiate into 3 cells called antipodal cells. So it's like a cell within cell. Just now it's a cell, the whole thing is a cell called megaspore, right? Now they continue to form cells there. It's called antipodal cells. Okay? And then another 3 will be over here. Okay? The one that is closest to the lubang is the egg cell. This one is the egg cell. The 2, okay, that is like a supporter like that, is called synergid cells. What is the meaning of the word synergid in English? It's like work together. So if I say, wow, we make a good team, you know, our synergy is very good. That means our kerjasama is very good lah. So synergid cells, they support the egg. Okay, let me repeat this part. So after 3 of the megaspores degenerate, the remaining 1 will grow. So after they grow, the nuclei, they undergo mitosis 3 times. So you will produce 8 nuclei. So you must know how they arrange. Antipodal means opposite pole. Because they're opposite to this lubang called micropyle. Okay? You must know how they arrange. Okay, do you watch football? Do you watch football? You don't watch football? Okay? Even though this is not the same number of players, but at least remember, this is what we call as the formation 3-2-3. I used to call this as formation 3-2-3. Okay? In this. So, you got 2 winger, 1 striker, 2 midfielders, and 3 defenders. Okay? So the main striker is the egg cell. The 2 wingers are the synergid cells. The midfield are the polar nuclei, and the defenders are the antipodal cells. Does it make more sense if you put it in that way? It's like a football match, you know? So the striker is the egg cell in the middle. They are right at the goal. This is the goal. Remember this as the goal post, the goal, right? So the egg cell is the striker. The 2 wingers are the synergid cells. And then you got 2 midfielders, the polar nuclei. and you got 3 defenders as the antipodal cell. They're at the opposite pole. Easy? So once this thing is formed, we call this as an embryo sac. So an embryo sac is something like this. So the later will become like this. Tom 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 3 2 3. And we call this as embryo sac. So where is the gamete? The gamete is this. Let us have a look at the essay. Okay, so this is just basically a picture showing you. So the important structure is this thing called micropyle. Can you see the micropyle? Micropyle basically is a lubang, okay, in the ovule. This is the ovule. You can see there's a lubang that connects inside. This is the micropyle. It's quite important for you to know. Okay. Yeah. So, pembentukan pundi embrio dalam ovule. So, sel induk megaspora yang diploid membahagi secara meiosis untuk membentuk 4 sel megaspora yang haploid. So, that's the first step, isn't it? So, 1 becomes 4 by meiosis. But 3 sel megaspora akan merosot. You can see there are 3 of them that will degenerate, left 1, right? So, hanya 1 megaspora akan mengembang, right? So, the 1, they become so big. You can see they become so big later. So the nucleus megaspora membahagi secara mitosis sebanyak 3 kali untuk membentuk 8 nukleus. Initially they are 8, right? So importantly, you must know how they arrange. 3 daripada 8 nukleus bergerak ke hujung sel untuk membentuk sel antipodal. These 3. This lubang is the micropyle. So these 3 are the antipodal. So 2 nukleus bergerak ke tengah dan membentuk nukleus kutub. This one. Nukleus yang paling dekat dengan micropyle akan berkembang menjadi sel telur, the egg cell. This is too small to picture, is the middle. Dan diapit oleh 2 sel synergid, the wingers at the side. So when after they arrange like this, pundi embryo dibentuk. So if we look at the English version, this is a summary. There are 2 polar nuclei. The egg cell is the middle. These 2 are the synergid cells. These 3 are the antipodal cells. I tell you first, this question a lot of students will confuse, which I don't understand why they confuse also. It's not difficult, but it's the same as just now, right? Microspore become megaspore. No degeneration, this one got degeneration. Mitosis 1 time, this one mitosis 3 times. Then you have to know the formation 3, 2, 3, 3, 2, 3. 3 antipodal because they're opposite pole, 2 in the middle polar nuclei. The one in the middle is egg cell nearest to the micropyle, and the 2 wingers synergid. That's all. What's so difficult? If you're okay, you are done, just write a letter okay in the chat box. If you've got a question, you can ask me. Okay? So, wow, today we studied a lot of stuffs, man. We have concluded the whole chapter 5, and then we also move on to, yeah, before mitosis is megaspore, you are right. Embryo sac is only after all the nuclei moved nicely. Then we call it as embryo sac. Okay? So today we learned a lot. We completed the chapter 5. We discussed all the exercises. And then we have moved on chapter 6 until the mid part. Next week, I think we can finish chapter 6 already. Do you know that? Next week, we can finish chapter 6 already. So, yeah, I will give the exercises this week. Please attempt them. Thank you so much for your attention. I'll see you guys again next week. Thank you. Bye-bye.